About 20 years ago, 3 groups of physician scientists reported a striking and coordinated expression of mRNA transcripts in the blood of lupus patients that had the common feature of being inducible by interferon. On close analysis, those transcripts were most consistent with an effect of type 1 interferon, the family of interferons that include alpha, beta, kappa, omega, and comprise our essential host defense mechanism against virus infection. The observation was soon supported by data from others and came to be referred to as the interferon signature. In 2021, the first drug specifically targeting the type 1 interferon pathway, anifrolumab, was approved by the FDA for the treatment of adult patients with moderate to severe SLE who are receiving standard of care therapy, an achievement that not only built upon those papers published in 2003, but on nearly 70 years of investigation. The research that led to the recognition that type 1 interferon is a critical pathogenic mediator in most patients with SLE is a terrific example of how careful study of patients can lead to better understanding of disease mechanisms as well as improved outcomes for patients. But it is also important to recognize the efforts of those who came long before we considered a role for interferon in autoimmune disease. Our review of the history of type 1 interferon research in A&R reflects our respect for the achievements of our predecessors, as well as the conviction that consideration of the groundbreaking research that builds our knowledge base over time holds lessons for all of us who aspire to gain insight into disease and improve patients' lives. In review of the early literature leading to discovery of the substance produced by virus-infected cells that interfered with a subsequent infection of cells with virus, we learned that while Isaacs and Lindenman are credited with that discovery, very similar observations were made several years earlier by a pair of Japanese investigators, Nagano and Kochima, who perhaps did not receive as much credit due to the in vivo system that they used and that yielded data that was more challenging to interpret. Then, through the efforts of Jan Vilcek, Sid Pestka, Taniguchi, and many others, the nature of the type 1 interferons, their distinct genes, and ultimately their mechanisms of controlling virus infection, as well as their effect on the immune system, were defined. We particularly highlight the outstanding scientists, including Gideon Schreiber, George Stark, and Jim Darnell, who pieced together the elements of the type 1 interferon receptor and its signaling pathway that were essential not only for development of an anti-IFNAR monoclonal antibody, but also the JAK family inhibitors. We also highlight the long road to identification of the robust interferon-producing cells, the plasmacytoid dendritic cells, initially described by Lars Ronblom and later defined by Fred Siegel, a clinical immunologist working in New York City, along with Youngjun Liu. I learned that while we often cite a 1979 New England Journal of Medicine paper as first reporting the elevated levels of interferon in patients with SLE and other autoimmune diseases, in fact, it was a Russian investigator, Simon Skirkovich, who first made that observation and even suggested that therapeutic inhibition of type 1 interferon might be beneficial. But we consider Lars Ronblom the rightful father of research on type 1 interferon as applied to SLE in view of his extensive and critical research on nearly all aspects of that family of cytokines and their role in lupus pathogenesis. Research is now focused on understanding why type 1 interferon is produced in a sustained manner over time, often for many years, in many patients with SLE, and how we can apply that knowledge of the role of nucleic acids and their receptors, such as toll-like receptor 7, for development of new therapeutics. 70 years of extremely productive research by dedicated investigators has brought us essential insights regarding the manner in which interferon orchestrates the response of our immune system to virus infection, but most relevant to rheumatologists, how we can use that accumulated knowledge to effectively target the interferon pathway to the benefit of our patients.